Good evening, Dr. Badhunamal, sir.
Good evening, sir. Good evening. Yeah.
Yes, sir. So, can we start, sir, now?
Yeah, sure.
Okay. I request Ms. Vanaja, ma'am, to give a brief welcome and hand over the session to Dr. V.C. Padmanabhan, sir.
Good evening, everyone. Happy to connect you after a break of session 13 by Dr. V.C. Padmanabhan, sir, Assistant Professor, Department of Bioengineering, NAT Agartala. A session related to the development of diagnostic against infection and antimicrobial resistance. And thank you, sir. Well, we welcome you for the session, sir. Session over.
Yeah, sure. Voice is audible, no?
Yes, sir. Yes, sir.
Sure. Thank you. Okay, thank you, team of faculty members from SEDI Institute of Technology. And today I will be giving an insights about the development of diagnostics against infection and antimicrobial resistance. And good evening to all the participants. Yeah. So today, as all we know that the medical sector has grown up into multiple folds, and the lifespan is taken care by the lot of advancements in the health sector. But in the earlier ages, it will be the health of any individual depends only on the age, sex, genetic factors and biological factors. But as of now, it is not only based on these factors. The individual lifestyle is coming into play. Social and economic networks are coming into play, and cultural aspects, political aspects, even the environmental conditions concerning is varying with respect to diseases and disorders. So as you know that the Sustainable Development Goal Target 3.3 advocates to end the epidemics of HIV, AIDS, tuberculosis, malaria, and neglected tropical diseases, and to combat the hepatitis and waterborne diseases and other communicable diseases. And it depends on the health depends on the health equity. The mental health well-being is an important factors which we have to consider, and environmental and social governance. And the future of medical science is not only about how far we are good in treatment. It also directly correlates with how fast we are in diagnosing the emerging diseases and emerging viruses, like emerging bacteria, the pathogenic systems. So how far we are good in diagnostics also a key important thing with respect to maintaining the health. So that is where the digital transformation and of all the assessments, all the diagnostic tools is happening in the past 2 decades, we can tell. And the challenges is adapting to the health system to promote the prevention and ensure the access and the care. So the development and spread of antimicrobial resistance is one of the serious issue which the World Health Organization and any health sector is facing, because you can, on the right hand side, there is a statistical point, the impact on the global health. You can able to see that in 2020 there is almost 5 million fatalities were there regarding the COVID-19, and 2021 it is almost 3.5 million. But you can able to see the AMR fatalities, it is almost 1.27 million annually it is happening. Okay, it is in the data of 2019 data. And the reason for such kind of an antimicrobial resistance is like the people live closer together, the changes in the climate and the land use, because now we have started to the deforestation is happening at a larger rate, and they are trying to convert into a tea plantations or coffee plantation units. So the wildlife is coming into the terrestrial areas, and where the human living space, and the zoonotic diseases are getting very easier to spread among the human system. So the changes in climate and land use is a very important factor, and more global travel and the trade, which is also one of the reason for these developmental in the AMR systems. And animals are more than just a food. So it is not like we are consuming only as a food. It is also act as a reservoir and vector for many different fungal systems or viral systems or bacterial systems. So these are all creating a very important challenge to understand about the bacterial infection or viral infection or fungal infection. thereby the next step comes as how we are going to, the system is getting resistant towards the antibiotics or the control agents what we are consuming. Further, how we can able to track this AMR with the very easiest route. Every time we cannot able to go and see the patients to understand how far the effect is. So we need to identify some other route by which we can able to track the AMR in a global level. That is one of the prime importance we are focusing now. Yeah. So here you can able to see the magnitude of rise. So in 2016, the AMR is around the 7 lakh death, and now in almost AMR expected in 2020 is around 10 million death is expected around. And and you can see in 2019, it is was estimated to be directly responsible for 1.27 million death globally, and it is higher than those from the HIV, AIDS, or malaria, leading directly to 1.2 million death and playing a role in increase up to 4.95 million deaths. So the thing is, like here, this particular graph explains that how much amount of a death is happening across the different regions as declared by the World Health Organization. You can able to see the associated with the resistance and the attribute to the resistance. You can able to see the differentiation over here, and most of the African countries is the top hits for the antimicrobial resistance or any kind of drug resistance, because naturally there the waterborne and foodborne diseases are very high. And further you can able to see the Asia, Eastern Europe, America, and finally you can able to see with the North Africa, Middle East, and Australasia. So this is the global wise distribution of death which is recorded regarding the drug resistant thing. Towards the end of this particular session, you will get an interesting idea about how these the developed countries are contributing towards this particular AMR. It is not about the control of AMR. I'm telling about how much they are influencing the AMR systems to the greater level. That we will see towards the end of this presentation. Yeah. So here you can able to see around the Asia is a major impact. Asia is having a major impact, and Africa is also having a major impact. So these two are the predominant contributors as of now towards the death related to AMR. But there is an interesting other finding also there. Where the developed countries like US, China, and Germany are contributing towards an extensive amount of antibiotic and its resistant entities, which is present in the common river samples. Yeah. So what about the targets for the global action to address? Yes, we know that AMR is not a regional issue. It is not only restricted only to India. It is not only restricted only to Tamil Nadu or Asian continent. It is a global issue. And just before COVID, World Health Organization has released an alarm note by 2017 regarding the impact of AMR. But what happened after 2020, as the COVID-19 has taken the upper edge, everyone has left about the AMR importance and other thing. And during COVID, you can able to understand every human being have consumed lots and lots of antibiotics and antibacterial control agents to a much higher concentration. Ultimately, all these antibiotics as well the control agents has released into the water streams through our fecal route, our through a urinary route and urinary tract route, right? So the extensive amount of resistance is experienced from the preliminary data, and the amplification is much higher than the before COVID and post COVID era. Okay, so the 10%, the recently 2024 Lancet has released control targets for AMR across across the countries, and the 10% of reduction in mortality from AMR, and it is also targeting 20% reduction in inappropriate antibiotic use in humans. This is another important area, and 30% reduction in the antibiotic use in animals. So in order to control the bacterial or a fungal infection, how far we are using these antibiotics without any control to the animal system in the veterinary sector, that is also a key important component which we have to target. And this is the target given by the Lancet to World Health Organization. By 2030, we have to have a different strategies to control the AMR in a much better perspective towards the One Health principles. So what are all the steps need to be taken? A massive global public awareness campaign has to be there. So every citizen, every living individual need to understand what is the importance of antibiotic or importance of anti-bacterial or fungal agents, and what is the limitations for using it across the different systems like human or veterinary sector. So whatever it is, we have to know what is the importance of this and what is the impact of this. So the second point is about the improve the hygiene and prevent the spread of infection. So it is not about every time we are taking a chemical substance as an antibacterial agent. What happens to our native immune property, that we have to enrich it so that the chances of consuming these antibiotics will get reduced. So how we can able to improve our native immune principles, that is also an important target by which we can able to extend the AMR infection rate. And the reduce unnecessary use of antimicrobials in agriculture, that is an important one. And improve the global surveillance of drug resistance in human animals. So this is a very crucial point, which has created a very important stroke in monitoring the COVID-19 spread during the COVID period. So earlier what they have done is after infection, they have started to identify the hub areas of COVID, but later stages through wastewater treatment plants, they have identified the major hubs of COVID-19. So for example, if a particular wastewater treatment plant is connected with the 10 different areas of a living system, populated areas, then if that particular wastewater treatment plant shows a higher concentration of these resistant genetic materials, then we can able to understand that these materials has been from those populated areas. So we can able to target that area to control the COVID. So the wastewater surveillance has played a very important role in identifying or we can call that as a forewarning in the COVID spread. And now it has been applied in many different areas to identify the spread of tuberculosis or AIDS or malaria. So like that, it has started to extend. Okay, so the promote the new rapid diagnostics to reduce the unnecessary use of antibiotics. So if we have a proper diagnostic principles, then the improper use of antibiotics can be reduced. So this is the two major areas where a lot of research scope is there, and the government of India as well, the World Health Organization, and many different agencies are there which are giving a lot of research funds for promoting their research on this particular area. So the ultimate endpoint of this is we are going to work on the One Health principles, where animal, environment, and human health will be taken care simultaneously. And in order to monitor that effectively, joint programming initiative on antimicrobial resistance and global antimicrobial resistance and surveillance system, which is shortly called as GLASS and JPI AMR, as is trying to collect all the data from the researchers, those who are working in AMR, and putting it in a common portal. Okay, so this is one of the important area by which we can able to extend our work on AMR related stuff. And the ultimate aim of this one is we need a cheaper, better, and faster test that can be used at a point of care to reduce the inappropriate antimicrobial agent use. So it need to be accessible, and it should be equitable, and it should be affordable for all classes of countries, because you can understand that developed countries, high income countries, middle income countries, low income countries, and research for life countries. There are many different classification of countries as per the World Health Organization and the research councils. So that we have to be, it has to be afforded. The methodologies what we are developing need to be afforded by all classes of the countries. That is another important one, and it should be eco-friendly too. So these are all the major points we have to consider when we are stepping into a antimicrobial resistance based research. And as I said earlier, the One Health response to antimicrobial resistance is about the how to reach or how to address the sustainable goals. And we have to be very clear with our objectives that human is not only the target. Human, the food materials, which in the terms of what kind of food we are consuming. So that food material also should be free of the antibiotics. Plants and crops, environment should be environment in the sense water and the air component, because many pharmaceutical compounds are volatile in nature. So air will also get affected, water will also get affected, land will also get affected. So such kind of things you have to keep in mind when you are developing a project work. And terrestrial and aquatic animals, which means the two important areas. where we are living and where we are dependent on. So those two things are very important. So that is, we don't have much time to start working on AMR. So it is always good to start working with the AMR in a multidisciplinary approach, because it is not only by the biotechnology researchers. A very important thing is required from the doctors, and another important stakeholder is the biotechnology researchers, and another important stakeholder is the civil engineers. Because whenever we are trying to use the water samples for the analysis, a perfect civil engineer will know. The civil engineer, in the sense an environmental engineer, with having a perspective of this one, can create early warning signs with respect to the wastewater treatment plants. And yes, to approach this, we cannot able to neglect the three important things. One is the international collaborations or national collaborations, and the open data sharing platforms. We have to, it is not like I am going to work, so I will not share my data with other fellows. It is not like that, because it is a many different factors are getting influenced by this. So we need to have a common platform where the data will be shared and we can able to work on towards a common objective. So that is the second major aspect you have to be very clear. And the third one will be, of course, you will be working on the research part. What are all the different pathogens are there? What is the priority list released by ICMR, World Health Organization? That you have to be very clear. And you have to get enough idea about the pathogens data also. So if you have a clear objective towards the AMR control, we cannot able to neglect the three important factors. One is international collaborations or national collaborations, open data sharing platforms, and your own personal research data. Okay, yeah. So as declared by World Health Organization and approved by ICMR, there are major areas which we have to target, and I have clubbed into four major aspects and two foundational steps. So the foundational step number one is like effective governance, awareness, and education. So now we are in this particular stage only. Now we are trying to educate the medical practitioners or medical professionals and the environmental engineers with the basic idea about the AMR, because they will not have a much clear idea about how fast these bacteria are getting resistance at the genome level. Right. So medical practitioners will have an idea about how to control this bacteria, and environmental engineers will have an idea about how to control this system, but they will not know how fast the antigenic shift or drift is happening in the genetic level in the microbial system. So that is where our role of biotechnologists plays a very important one. The second important foundation step is the strategic information through surveillance and research. So now, after getting a clear kind of an awareness and the governance and the basic education, then we will step into the research work, which is focused on four pillars. One is the prevention. Second one is access to essential health services, timely and accurate diagnosis, and appropriate quality assured treatments. So these are all the four major pillars by which we can able to develop an effective surveillance as well as a treatment for the AMR. Right. And the timely and accurate diagnosis, out of these four pillars, this plays a very important one, and we are stepping into the research on this particular area. Okay, so coming into diagnostics, the timely and accurate diagnosis is very important for allowing an active surveillance of the drug resistance. We need to know because the microorganism may develop a resistance against first generation, second generation, third generation within a span of 2 months or 2.5 months of a time. So we need to have an effective diagnostic procedures which can give you an answer at a very faster rate, so that the extensive research can be completed within that short period of a time, so that you will get a yearly warning signals again from the common ecosystem of aquatic life. Okay, the second one is about enabling the optimum use of existing drugs. Yes, if we know the diagnostic procedures very clearly, then we can able to tell that to control this particular work, this much a drug is not required, only this much drug is required. Instead of that, now they are giving the maximum. Now the prescription is based on the maximum dose of anything. Okay, so if we know the how much is the infection rate with respect to the system, then we can able to give how much amount of antibiotic is required for the control. So that is a very important thing with respect to the diagnostic procedures. And the third one is about protecting and ensuring the appropriate use of the new treatments. Yeah, new developed treatments are also an important factor which we have to consider, because as I said in my first session, to develop a drug, it may take 5 years or 6 years of a time as of now, but the resistant development by the microbes will takes place within a month of a time. So that is where that you might have experienced in the COVID time also. The variant number 1, variant number 2, variant number 3. So many different variants has come within a period of 6 months or 7 months of a time. So that is a very important one where we have to work on the diagnostics. So diagnostics is not only done by the biotechnologist. An effective electronic engineer or electronic and instrumentation engineer is required. So whenever you are planning to write any research proposals, you try to form a group of professors or scientists who are having expertise in biotechnology, uh, uh, electronics and instrumentation side, so to develop a diagnostic kits or to develop a diagnostic sensors. Okay, and three major ways that diagnostics are used for AMR is to guide the patient management, to reduce the overuse, and infection prevention control is very important one, and the surveillance based. Okay, so where do we need diagnostics for AMR? So now you will think that, okay, I have developed a sensor. Who will be my customers? Who will who will be looking for my developed product? So that is one thing we have to target, because without the audience or without the application, we will not nowadays the funding agencies are not promoting the research work. So we need to have an applied research in a much broader way. So at community level, as home test and self testing or self sampling, so at a community level we have to have a diagnostic sensor. That can be an important area. And a primary care level, a simple rapid test to distinguish bacteria causing fever, respiratory and enteric infection. So it is not like just an entry of bacteria is different, and a bacteria which is having potential to cause respiratory infection is different, and enteric infection is different. But now we are consuming a same kind of an antibiotic to control all the three. Okay, so that is unnecessary. So at a primary care level, it will be a much better way. And a test that can identify a pathogen and its AST, antibiotic susceptibility titer, will help you to give a much better view. So here you can able to see the World Health Statistics of 2024, the health worker density and distribution, the medical doctors and the nursing and midwife personnel and dentist and pharmacist. Okay. So what are the opportunities through new diagnostics? You can able to target your research through handheld lab on a chip. It will be like a small sensing kind of a device where you can able to understand. If you load your sample, you will get a clear idea about what kind of a gene is there, what kind of expression rates is there. So that can be done. And second one is the point of care testing, where the C-reactive protein will be taken care. Right, if an infection happens, the C-reactive protein will be shooting up in our blood level. So based on that can be used as an identifying markers through microarray you can able to do. And the last one is about the high throughput genomic technologies, where the whole genome sequencing or metagenomics will be used to do the surveillance. So these are all the three different major areas where you can develop your research proposals towards the diagnostic sectors. And as I said, it is another consolidation I have given over here. The conventional method is the PCR or manual antibiotic susceptibility test, which is a very old method. And now the non-conventional method is the metagenomics and multitope spectroscopy. And microfluidics based will be like your point on a chip that can. Okay. And this here I have given the cultural dependent methods. That is a conventional method. You can able to see, it's very simple. If you want to understand how your bacteria, what kind of a bacteria here there, first what we will do is the earlier method, infection will occur. Then we will take the patient sample, right? Then we will see the antibiotic susceptibility tested or a titer conducted on the sample. Then we will go for the therapy. Then the patient will respond to the treatment. This is the earlier method to treat the pupil. Okay, but now it is not like that. We have to develop a method which is very rapid to identify within a day, and then immediately we have to stop the proliferation of the cells. And here you can able to see the differences in the Haas. So earlier it is like a 36 to 72 hours will take to get the results, and rapid test will give you 30 to 40 hours, and now it is within 8 hours of a time. So now all the medical sectors are looking for the diagnostic devices or assay testing kit, or with which can a rapid test kit which can give you the results within a shorter period of time. So that is where you can develop the new novel technologies to combat the AMR. And the other one is about the molecular based. Yes, you know that based on the genetic material, we will try to identify the resistant genes, and we will try to incorporate how we can able to use it in the real-time application. So it is again, it is PCR based, and you know very well about the conventional PCR and how it is done. And here it is another important area where we are working on right now, and it is a whole genome sequencing or metagenomic platform by which we can able to identify what kind of a bacteria is prevalent in this water, or what kind of a system is there. So you can able to see the diagnostic samples are there. From the results, we can able to identify what are all the different kinds of pathogen, what are all the AMR is there, and how we can able to assess the distribution patterns, what are all the different bacteria are there. So all these informations can be identified through the real-time surveillance data also. So one case study I have given, which we have done in our lab, so that I will be giving in the next two to three slides. Yeah. So first, it is very simple. We have to collect the pathogen sample. We have to identify the pathogens through the whole genome sequencing and compare the genomic sequence. Then you can able to identify what bacteria, what is the intents of the bacteria. Then you can able to prescribe the antibiotics. Then you can able to understand what kind of a methodology I can go for the treatment plans. Okay. So there are certain databases for the advanced level genomic studies where the pathogen watch, finding phenome, EMBL or CARD database, Magnify, NCBI, Uniprot, KEGG pathways, so KEGG databases, so and the metagenomic analysis server and the NCBI, all these things are very important databases by which we can able to identify the advanced genomic analysis concerned with AMR. And the GLASS and the as I said earlier, JP-AMR is one of the important one where you will get an enough amount of data on AMR with respect to the global research. So microfluidics, as I said earlier, it will be like a small kit kind of a thing, like a lab on a chip. You can able to see the sample preparation will be over here where you will be loading your sample. This will be the reaction site. This will be the result observation site. So if it will be traveling through, it will be just like a pregnancy kit. Okay, so when we load a sample, the data can be identified through the difference in the color, and based on the intensity of the color, you can able to understand what kind of how much is the load of these antimicrobial resistance is located over there. So this is one kind of emerging research area where we are trying to develop the small kits towards identifying different resistance and different diseases. Yeah, so this is what the important area where we are working on. We are trying to identify the resistant diseases and the resistant entities through the wastewater surveillance. So we can able to identify the infection through wastewater systems, and it can be done within a period of 2 to 3 days of a time. We can able to identify, right? So this is one of the important thing which we have to concentrate in the next 2 to 3 slides. Okay, so the case study from our group is we want to identify and monitor the fluoroquinolone. It is one of the important antibiotic which we have seen in the first session also. We worked on degrading these fluoroquinolone. So we want to identify how far the fluoroquinolone resistant bacteria and genes are there, and which is creating the antimicrobial cluster. So to give a rough idea, this is the core structure of the fluoroquinolone, and the mechanism of action of this antibiotic against the bacteria is it inhibits the DNA synthesis of the bacteria by disturbing the topoisomerase 2, DNA gyrase, as well as the topoisomerase 4. So these are all the three major targets for the bacterial system. Okay. And how the bacteria is developing resistance against this fluoroquinolones. So there are three major possible ways. One is called target mediated genes, or by plasmid mediated, or the chromosomal integrated resistance. Okay, so the target mediated genes, the four major genes are important: the gyre, gyre-B, PAR-C, PAR-E. Plasmid mediated is QNR and AAC, and chromosomal OMP and QEP, NOR and OQX. Okay, so these are all the major resistant genes which contributes towards the resistant mechanism of bacteria against the fluoroquinolone. So if these genes are, because if the bacteria is getting lysed, these genes will also be released into the system. So as a result of that, the wastewater will have these kind of a genes in a prevalent way, which shows that the resistant bacteria is very high in this particular water. So that is what the hypothesis what we are trying to put forth here. So the importance of each resistant genes is given over here. So you can able to understand all the major important enzymes for replication of bacteria is the target. Okay, so DNA gyrase or topoisomerase 4 or topoisomerase 1, porins or membrane related one, regarding the active and passive pumps. So regarding these are all the major sites by which the bacteria is developing resistance against the fluoroquinolone. So these will be my targets. So what we have done is we have identified by till October 9, 2023, we tried to identify what are all the bacteria which is having the these particular genes, because in our study we are going to target only on this target mediated genes. We are not concentrating on plasmid mediated and chromosomal mediated. In this work I am saying, so our target is only the gyre B and PAR-C, PAR-E. So these are all the two major targets. Okay, so here from this we have identified what are all the bacterial families which are having these particular resistant genes in a larger quantity. And there is a very interesting information, and from the list of bacteria what we have collected, those bacteria are showing this kind of a relation across the 4 different areas. One is the gram staining. So out of those bacteria, 76.63% of bacteria comes to gram negative, and 23% belongs to gram positive, and all the bacteria are supporting for the formation of biofilm. If the biofilm formation is very high, then the resistance development will be very high. That is a correlation we have. And the siderophore formation will also be very high. And 5.19% of the isolates are spore forming isolates. So here you can able to see the correlations of all these bacteria which are hosting these resistant genes, right? So here you can able to see the phylogenetic correlations between the FQR microbial isolates. So the 3 different clades are there: clade A, clade B, and clade C. So 5 phylum, where the 8 class bacteria of 5 different phylum, 8 different class, 18 order, totally 77 species we have worked on it, and which we are locating into 3 different clades. One is clade A, it is proteobacteria, and clade C is Pyrrhocata, and clade D is the combination of Bacillota, Actinomyceta, and Bacteroidata. Okay, so these are all the different kinds of organisms coming under the different classes which are having the resistant entities against a fluoroquinolone antibiotic. Okay. Yes. So then what we have tried is we wanted to try in the global scenario how the gyre genes are distributed. Okay. So we have identified the methodology to shortlist how the gyre, gyre B, and PAR-C, PAR-E are distributed. So here you can able to see the A gyre. You can able to see this one. Okay. The representation A to G, which is gyre only, gyre B only. C represents both gyre and gyre B. D represents only PAR-C. E represents only PAR-E. And F represents both PAR-C and PAR-E type of genes. H represents all gyre. I represents all PAR-C, including unclassified. And G represents all. Okay. So here you can able to see the heat map where the 77 different bacteria are listed over here, and the resistant entities are also listed over here. Here you can able to see the extent of resistance is very high with respect to the green signaled one, which is nothing but E. coli Shigella and Salmonella enterica and Klebsiella pneumonia and Campylobacter, Clostridioides difficile, and Neisseria gonorrhea, and Staphylococcus aureus. So you can able to see the relative distribution of these particular genes in the different set of bacterial system. Okay, so this heat map gives you an idea about what are all the bacteria which host these resistant genes in a very higher rate. So we wanted to understand how these systems are prevalent in the living water streams. So based on that, we have identified the spatial distribution of these 4 genes, because we are targeting on 4 genes. One is gyre, gyre B, PAR-C, and PAR-E. So here you can able to see the America is hosting almost these resistant genes are highly prevalent in America, were reported, and China also. And the next one is about the German. So 3 important countries are highly reporting the resistant genes. It doesn't mean that Africa and rest of the Asia other than China are having the lower content. these countries are reporting the resistance in a much faster rate, and immediate diagnosis practices is there, and they are reporting to the system. And still we are in the primitive stage to report as a AMR, and the studies on AMR is very primitive across the other countries and other low middle income countries. So from this, here you can able to get the relative distribution everywhere in all the 5 different types of regions, because this regions has been classified as per the World Health Organization: North and South America, Asia, Africa, Australia, Oceania, Russia and others, and European countries. So here you can able to see the relative distribution of all the genes across the system. Here you can able to see in the North and South America, especially in Colombia, the PAR-E gene is very high. The distribution of PAR-E gene is very high, where PAR is very less, right? But on this other side, if you can see in the African countries, in Kenya, PAR is very high. Cameroon, PAR is very high. Africa, PAR is very high. But Uganda and Ethiopia, the PAR-E is very high. So this is how we can able to understand what kind of a genes is very highly distributed across the countries and across the different ecosystem. So that made us to identify from this graph. We identified that 3 important areas are the main targets. One is America and China, as well the German. So we have identified the major rivers which are flowing across these countries. So in USA we have taken Mississippi, Missouri and Arkansas, and German we have Danube, and Japan we have taken Tone. So these 3 areas we have started, and to China also, it is mentioned over here as Yangtze, Yangtze, Yellow and Pearl River. So these 4 rivers we have taken into account, and we wanted to study how far this resistant genes are distributed in the water streams. Okay. So here you can able to get the distribution of water fluoroquinolone antibiotic in the river streams. So from the river Mississippi, these 5 antibiotics are reported at the range of this much milligram per liter. This much ppm it is getting reported, and across the rivers we have collected the data, right? And the ecological risk assessments was done. Here you can able to see the ecological risk assessment is very high with respect to these antibiotics, especially in the Yellow River of China. Okay, so enoxacin, synoxacin. So to the first generation of antibiotics is highly reported in Yellow River across the different categories of living systems across the ecosystem: fish, daphne, green algae, in terms of acute toxicity and the chronic toxicity we have assessed. So the ecological risk assessments was done with respect to these 2 classes of this particular rivers of our consideration, because these are all the hotspot regions according to our study. And this is for the experimental toxicity value. You can able to see the ciprofloxacin, norfloxacin and enrofloxacin with respect to the Yellow River. The earlier one with respect to the predicted acute and chronic, this is the experimental one. So both the prediction as well as the experimental one are very close, and Yellow River is showing the much higher concentration of the resistance. Okay, so the mixed risk coefficient we wanted to study, because these in the living system or in the ecosystem, these will not be available in the standalone items, right? So you will be having the afloxacin class, norfloxacin, ciprofloxacin, many different systems will also be there. So we wanted to study how it is behaving in a real-time situation. So we wanted to study the mixed risk coefficient, where the in the experimental toxicity, Yellow River stands a very important one, and in the here also the Yellow River stands at the third one. Yellow River stands at the higher one. So in the predicted as well as in the experimental one, I think Yellow River, and here also Yellow River. Both the cases Yellow River is showing the higher risk coefficients. Okay. So this is the one important thing by which we can able to understand how far these antibiotics and the resistant genes are correlating with each other towards the in the native streams of water. Okay, so now we wanted to have an interesting features. We wanted to study what will be the effect of these metal ions onto the resistance, because here you can able to see the A, B, C, D, E, F, G, H, I till I. It is represented as the different classes of the genes. Okay, and carbonate, sulfate, chloride, nitrate, calcium, magnesium, and sodium and potassium. So these are all the different ions which are prevalently present in the drinking water or in the river streams or in the lake water or in the aquatic ecosystem. So we wanted to study how far it is closely related, right? So we got a very interesting information that all the ions are playing a very important role in generating the resistance. So here you can able to see the K plus is having a very high correlation of 0.595, and chlorine is having a very high relation of 0.628 towards the resistant development. So it is not like the appropriate quantity of chloride and potassium is there. If excess amount of this there, the bacteria will try to develop through the efflux pumps what it is happening to develop the resistance. So that is what the correlation which you can able to understand from the Spearman analysis here. And we try to study how far these are interacting together regarding the genes and the ions. So this particular graph will shows how far the correlation is between the resistant and genes as well as the metal ions through the Spearman correlation. It is very closely interconnected. All the ions are having a very close interaction with the resistant genes. So this is a one kind of a primitive study which we have worked on the antimicrobial resistance. So the take-home message from this particular lecture is, yes, if we are not properly taking care of the antibiotics, this particular study is a proof that it also get released into the drinking water sources also. And through when we consume the drinking water sources in a different routes, whether it is a treated or untreated, naturally higher possibility of are there to get into our system. So naturally, through the recombination systems, the natural microbiota which is present in our system also are capable of developing the resistance against the antibiotics. So this is one of the important one we have to consider, and wastewater surveillance is a very important area by which we can able to go with the early diagnostics at a shorter time. So once again, I would like to thank all my funding agencies and collaborators who have helped across to execute this kind of a work across the different countries. And for a special note of thanks to DST and DBT for funding this project work. And thanks to my research group members also for making it more effective regarding the antimicrobial resistance and allied works. Yeah, thank you. Thank you for the time.
Thank you, Professor Batmanathan. Yeah. So now the session is open for discussion. Participants, if you have any question or query, kindly let us know in the chat as well.
Yeah.
We'll have another 5 minutes for discussion. If you have any query, please let us know in the chat.
I think any in chat box, any question is there? Okay. So any kind of a queries if you have. No, sir. The PPT cannot be shared actually because it have a lot of data which are funded by DBT. So we will try to see whether if it's possible, I will share that. Yeah.
Professor Batmanathan, if you could clear if any mode of early detection of the presence of antibiotics in the ecosystem, is there any currently available or commercialized product?
No. In India, it is in the early stages of research only. And through wastewater survey or water surveillance or any kind of a thing, it is a emerging area only not only in India, because this wastewater surveillance has picked up only during the COVID time. By 2022, 21 to 22, it has emerged as a very strong surveillance unit. So it is in the early stages of research only. So it is now many papers are coming and many routes are identified. They are trying to optimize the process by which we can able to get the optimum concentration of these antibiotics. Antibiotics is there, but resistant genes, it is not much. Researchers are not going on that. So that is in the early stages of research only. But antibiotic residual concentration, it is very, it is known for more than 2 decades of time.
Okay, sir. So in your answer, with your answer, I would gather that, or I would understand that if any researcher focus on the research of antimicrobial resistant gene prediction or detection, that would be, I mean, they can start even now in India.
Yeah, yes.
At this stage.
It's always welcome to start the work on immediately. That the thing is, like, many different sectors are there to work against AMR. Antimicrobial resistance, a class of researchers will be trying to develop a new drug or new antibiotic against the bacteria. That is one kind of research in AMR. The second one is how we can able to develop the diagnostic kits to identify the changes in the resistant development of a bacteria. That is another important area. Third important area, how to identify the concentration of antibiotics in the streams, river streams, or water treatment plants, or it can be your food sources, your drinking water sources. So that can be another possible area. And another probable area will be the how your resistant genes is developed. So that will go much deeper into the genomic area. Genomic side of assessments will be there. So that can be another promising area where you can extend your research work. So many different areas of focus are there in AMR. It is not only about the wastewater surveillance is there. But the thing is, like, for any kind of an AMR application, it is not only a biotechnology engineers alone cannot do the job, because otherwise if we are targeting only through biotechnology people, it will stuck at one point of time. So we need civil engineers, we need electronics and instrumentation engineers, and we need a proper basic science engineers to get into the real-time application towards the customers or government bodies or government systems or any kind of a systems. So that is one thing you have to keep it in mind whenever you are developing a proposal. Otherwise, it will be like only a basic level of research, it will be there.
Understood, sir. Thank you. Thank you so much for your input.
Yeah.
So participants, if you have any query, you may ask now, last 2 minutes.
Yeah, you can unmute and ask if you have no issues. Sir, the consideration, will you share the PPT? The thing is, like, I think I can share without excluding my case study data. I can share the details, no issues on it. Because since it is a funded project from a national and international collaborators, we cannot able to share it without their permission. Just for popularizing these things, we can able to do it, no issues. But we have to get a proper permission from the agencies also to it. So I think I will share the first half of the PPT. Definitely I will share, no issues on it. Yes.
Right, sir. So we come to the end of the session.
Yeah, thank you, Dr. Amal and Vanaja, for your time. And if any queries are there, or if any kind of support any of the researchers are requiring regarding the AMR, definitely our team is ready to help in terms of suggestions or in terms of collaborations or whatever it is. We are ready to support. So you can note my number as well as the email ID. So you can contact through both. Anything is fine. So whatever maybe the support we can able to give, definitely we are ready to for the friends and all. Yeah. Thank you.
Sir, Dr. Brinda ma'am is
yes, sir. Actually, recently we are just working on a diagnostic kit for PCOS, sir.
Okay.
For PCOS using biomarkers and saliva, like a portable kit which is affordable price. So what is your suggestion about that, sir? How is it? Is it like what can we way it can help us out? We are just working on it. We are planning on it.
You are working on the PCOD related, no?
PCOD, yes, sir. My major is molecular biology and genetics.
So that is that sector is a different one, because the developing sensors for different diseases and disorders
Okay,
to
sir.
early, that is a different picture. That is because that doesn't have a much impact on antimicrobial resistance and other things.
Okay, okay, sir. Okay.
So that can be done. That is a very prominent area which many people are working on it, because
Okay,
whatever the diagnostic kit you are looking to develop,
Yes,
it has to give the results in a very shorter period of time. So earlier it was like 3 hours of a data, but now it is coming around 1 hour of a data.
1 hour.
And
Exactly, you're right, sir. 1 hour, like, yes. First it was 1
the,
and 10. Now it has become 2 of 1, the ratio.
yeah, now the market will be looking for some other diagnostic kits which can be finished within 10 minutes of a time. So everywhere the work on the diagnostic sensors
Okay,
will
sir.
always be targeted towards a shorter period of time. So
Okay.
that in that aspect, the electronic and instrumentation engineers will play a very important, crucial role, because they know how to identify the sensors which can give you the results at a very shorter period of time. So only if bioengineers are working on it, it will take a little bit more time, because we will be more targeting on the conceptual side. But
Yes.
these instrumentation engineers will also know what is the kind of a sensor which can be used for the early detection in terms of potentiometric or in terms of amperometric. So they will have that kind of a knowledge also. That can be the amalgamation of these two will give you a better results in a shorter period of time.
Okay. Sir, coming to like DRDO or our defense project, no, sir, DRDO is
Yeah.
anyway this diagnostic, like development of diagnostic against infection or anti, is anyway this will be helpful, sir.
Yes,
Actually,
ma'am.
now DBT has opened out a scheme for DRDO to write at proposals for the persons who are working in military. They are ready to pay even to 1 crore, sir.
Yeah,
But
yeah.
it has
So
to work accounts, yeah.
as of now, immediately ICMR one call is open,
Okay, sir.
which is funding for these kind of diagnosis, disease treatment, or drug development like that. I think
Okay.
now
Oh, let
and
me check
in
out, sir. Okay, okay.
India, so it is already there. They
Okay,
are
sir.
looking for the diseases which are associated with the low temperature disorders or diseases.
Yes.
So
Okay.
the diseases which are prevalent for the military people. So that they are looking for. Past 2 decades they are looking for. So then, and many researchers have also given many different finished ideas to DRDO submitted. That is also there. So it is one of the important area where we can pitch in, because there, when you are trying to apply for DRDO, you have to think one another thing. The
Yes,
kit
sir.
which you develop need to work in that extreme conditions of temperature.
Yes, sir.
That is the only.
They have given us a scheme out, sir, around 6 to 7 places. So that's what, after listening to your talk and everything, I have come out like maybe we can work on this aspect part
Yeah,
and write a
yeah,
proposal of 2 page. Yeah.
yeah. Because only they will be targeting on whether it is feasible at this extreme of conditions. So
Exactly,
if
sir. Exactly.
your developed procedures or protocols or the isolated enzymes is working very well in that particular region, then it is always fine. Now the diagnosis is coming not done at the in situ level. So
Okay.
if you can to make it in the on-site level, it is always welcome. So that
Okay, sir.
definitely it will be much helpful for the defense personnels.
Sure, sir. Sure, I'll be in touch with you, sir, regarding if this is being continuously working on, maybe I can work out a framework.
Sure,
And
sure.
if so, I'll sure be in touch with you. Thank you, sir. Thank you so much.
Thank you. Participants, if you have any questions, you put in the chat or you unmute yourself. Last few minutes we can have with Dr. Padmanabhan. Okay, we are at the end of the session. Thank you, Padmanabhan sir.
Thank you, Dr. Amal, for the time you have given to share our experiences in AMR, and all the best for all participants towards your research work. Have an interesting research again. Thank you. Thank you, everyone.
Definitely. You
Thank you,
enlightened us on the AMR relevance through 2 sessions. Thank you so much for your interest as well as your time, sir.
Sure, sure. Thank you. Thank you so much.
And participants, I request you to fill up the feedback form. And we are come to the end of the session. Probably by 7 o'clock, you will have the test. I already mentioned it is to be taken in the ATAL portal. You may log into your login ID and you can check out on the left side. You'll have the assessment. Total, we have 30 questions. Each question carries 2 marks and you have 1 hour of time. So kindly complete the test. And attendance has been already posted. Still, today's attendance only need to be posted. So we will post it soon now. And we request you to join the validate function, validatory occasion. We'll let you know in the WhatsApp group regarding the time. And we are really grateful for your valuable time you spend with us. We meet you in the validatory session. Thank you. Now you may leave. Feedback link is there in the chat box. Complete your feedback. Thank you.